Hello viewers, welcome to Biodrunks. I'll be providing notes, PDF link in the description.
Let's start this class with lipid bilayer. So, what is lipid bilayer? Before knowing that, let's quickly understand what is this lipid made up of. Okay? And what are the different types of lipids and how it forms a bilayer. Okay? So, lipid is of three types, as you can see here. Okay? Now, this lipid, when it is linked with a phosphate, it forms phosphate plus lipid, which is phospholipid. If it binds with a carbohydrate, it forms glycolipid, and one is steroid. Okay. So where do you find this phospholipid? Phospholipid, you find it in the lipid bilayer, or if I should say in more detail, it's in the plasma membrane. Okay. There are many other. Plasma membrane is one. Plasma membrane. Okay. In one of the video, plasma membrane is already dealt, in which the detailed explanation of lipid bilayer according to the plasma membrane is already given. Then comes glycolipid. So this glycolipid, it contains what? A carbohydrate and a lipid. It's of two types. The simplest glycolipid is psergoside. These are derived either from single sugar. It is derived from a single sugar. Whereas this gangliosides, they are complex glycolipids and they contain a branch chain of 7 sugar residues. Okay. So these contain a branch chain of 7 sugar residues. Now these are found mostly in the outer membrane of the plasma membrane. Then you have sterol. Sterol, what is sterol? Sterol is the basic structure of steroid nucleus. What does it contain? It contains 4 fused rings. Okay. And this ring is made up of 3 6 carbons and 1 5 carbon. Okay, 1 with 5 carbon. They give a very rigid structure. This cholesterol, which is only present in animal cells, it's very important for the plasma membrane. Then you have cetosterol, sigmosterol, which is present in plants, and agrosterol in fungus. So this was about the types of lipids. Now let's go with the main lipid, that is phospholipid, which forms a lipid bilayer. Now you learned about it. So now you know where this lipid bilayer came from. How is this lipid bilayer formed? It is formed of a hydrophilic head and a hydrophobic tail. So this is your hydrophilic head and these are hydrophobic tails. They are 2 in number. This head is made up of alcohol, phosphate and the glycerol. Okay. And this has 2 fatty acids with it. If you know, hydrophilic means water liking and hydrophobic means water hating. So that is why these tails are present inside and this heads, they are present outside. For example, if this is the cytoplasm, okay, on the side of cytoplasm, the heads are present and then this is your outer side of cell or exterior. Okay, that is where your head is present. But what about the tails? They are present inside, in the core. Okay, because they hate water. Okay, so now you know what is a phospholipid. Now, this phospholipid is made up of either a glycerol or a sphingosine. If glycerol is present, it is called as glycerophospholipid. And if sphingosine is present, it is a sphingophospholipid. Okay, so this glycerophospholipid, okay, it has different, it contains a glycerol, fatty acid, phospholipids, and alcohol. Okay, if the alcohol is serine, it is phosphatidylserine. If the alcohol is choline, it is phosphatidylcholine. If the alcohol is inositol, it is phosphatidylinositol. And if the alcohol is ethanolamine, it is phosphatidylethanamine. Okay. Now, sphingolipids, they contain an amino alcohol. What is it? Sphingosine. Okay. Instead of glycerol, they contain this. Okay. So, in the sphingosine, sphingomyelin is one which is most abundant sphingophospholipid. Okay. Now, this phospholipids. Now let's find out what happens at neutral pH with this phospholipids. At neutral pH, you have three different charges. Okay. First is no net charge. Okay. And then you have a negative charge. And then you have a positive charge. Isn't it? So when I talk about no net charge, in the no net charge, you have phosphatidylcholine with no net charge and phosphatidyl ethanoamine with no net charge. So they don't have any charge at a neutral pH. Now, what about negative charge? So, negative charged are phosphatidyl inositol, which is negatively charged, and phosphatidyl serine. Okay. Rest, you have positive charge elements are very rarely present. Okay. So, till here, we understood most of the topics. We started with what are the different types of lipids, and then lipid bilayer, that is what concludes to our topic, what is lipid bilayer, and then how it is a different. If the lipid has your glycerol, it forms glycerophospholipid. It has sphingosine, it forms sphingophospholipid. Now let's find out some more characteristics about lipids. So these lipids are either saturated or unsaturated. So when I say they are saturated, that means they don't have any double bond. So let's understand that structure in detail. Why? And you could easily answer these questions. You can remember it for life. Okay? It's really very easy. Let's write it here. CH2, 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 CH2. Let me just write it randomly. So it has all single bonds. That means no double bond is present in saturation point. Okay. If I go for unsaturated, you will find double bonds in it. Okay. So these double bonds, how does they affect the structure? Okay. These double bonds. See, this does not have any bending. How? You can see it here. Here, this is your structure. Okay. So now this is how the tails are, right? So you cannot find any bending here. In the same case, if I draw it here like this, imagine this is a double bond. So knick is formed. Again, this is a double bond. So another knick is formed. Here, I will be joining another one. Okay. So if you can see it, how properly they are arranged. Okay. But what about this? See here, this is a neck. So will come out. This is where the disbalance occurs. For example, this also has a double bond. These are unsaturated. So if you can see, they are not properly attached to each other. Okay, so here you cannot find bending, but on the other case, you can find bending, that is in saturation. So here you can easily answer it, right? Which is a stronger bond and which is a weaker bond. If I say which is a stronger bond, it's obviously one with the saturated one, and this is a weaker bond. If you can see here, these are not being attached properly. So that means it is a weaker bond that is being formed. When the bond is strong, okay, you need high temperature, right? That is high melting point. And when you have the melting point high, the fluidity will be less. So easy, isn't it? Saturation is very easy. Because you don't have a double bond, there is no bending, and it is a stronger bond. And because it is a stronger bond, the melting point is high, the fluidity is less. In the other case, in the unsaturated, because the double bonds are present, you can find a knick or the weaker bond, and because of which there you have a low melting point. And if you have low melting point, those things can move easily, that is fluidity is more. Okay, let me give you a very simple example. For example, you are here present in a temperature of 45 degrees Celsius, and again you are being sent to a temperature of minus 5 degrees Celsius. So let's talk about two cases. Okay, so in the first case, what happened? You are being sent to this temperature. That is what your body should. What happens when you go to polar places? Everything gets solidified, isn't it? So that means here you need to have fluidity. So to maintain fluidity should be more. To maintain more fluidity, the melting point should be less. There already the temperature is very low, and if you have the melting point very high, then the fluidity will be more and you cannot move. That means here you should have unsaturation acids. Because if it is unsaturated, you will be having a low melting point and the fluidity would be more. Otherwise, what will happen? You will get stuck. Your blood won't move, isn't it? In the other case, let's go to the other case. When you are coming back from a low temperature to high temperature, it should be a saturated one. Okay. Why? At high temperatures, the fluidity is more. If more is the fluidity, that is also not good. That means I need to reduce the fluidity. That means I should have a high melting temperature. And if I'm having a high melting temperature, that means my fluidity is less. I think it was very clear. Yeah, and it was actually very easy. See, these lipid layers, that is, here it is where you have the link. Okay, if you can see this, if they don't attach properly, okay, what will happen? You will move, it would move, okay, because it has to move like this, isn't it? So if the temperature is very high, okay, what will happen? It will move more fluidity, the bonds will become theta like this. We don't need that, we don't want that. So what are we doing? We are increasing the melting point, and the melting point will increase, the fluidity will decrease. That means if it was moving like this, now it will move in a slower pace, isn't it? So that's how it is related with saturation. Now same thing goes for cholesterol also. That is, if you have a high cholesterol, that means it reduces the fluidity. What does it do? It reduces fluidity. Okay. And at high temperature, it needs high temperature for this. High cholesterol, high temperature to burn. Low cholesterol, already the cholesterol is low. That means you should have a low temperature. And what does it do? It increases the fluidity. Okay. This was quite easy. Now the next part, what we are talking about is the way your lipids are present. Okay. So as you can see, if the total lipid is 50 from here to here, if the total lipid is 50, okay, most of the sphingomyelin is present in the outer. Let me write it here. This is outer side and this is the inner side. So I've written inner leaflet and outer leaflet. So here, outer side, you have more of sphingolipid, sphingomyelin. Same here goes with the phosphatidylcholine. Okay, so it's present more outside and it's present less inside. And again, when I say about the next one, that is phosphatidylserine. So as you can see, there is nothing outside. Everything is present inside only. Okay, more, its concentration is more inside. And the last one, ethanoamine. It's nearly like some part outside and some inside, but more is inside. More it is towards inner side. Okay, so this is the main part of lipids. So hope the lipid bilayer is too much clear and it is very easy.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.